Welcome all of you. This class we are going to discuss about soil fertility evaluation. Soil fertility evaluation. It is a important approach to find out the nutrient supplying power of the soil. How much amount of nutrient, how extent the soil can supply the nutrient can be identified through this evaluation. So the optimum productivity of any crops depends upon the supply of plant nutrient from the soil. And the quantity of nutrient required by the plant depends on different factors like plant species and variety, yield level, soil type, then environmental factors such as climate, water, temperature, sunlight, and apart from that, agronomic practices. What kind of practices we are following? Based on that, the nutrient quantity of nutrient required can be calculated. So after analyzing the soil fertility, after evaluating the soil fertility, we can give the fertilizer recommendation to the farmer. Then the farmer, according to that, the farmer will apply the required amount of nutrient to the soil. There are different methods to evaluate the soil fertility. Visual method of diagnosis, like nutrient deficiency symptoms. Plant will exhibit certain stresses through the symptoms that due to nutrient deficiency, and analysis of plant. The plant analysis, like a whole plant or plant parts of growing plant, can be obtained. So that also giving indirect indicator of the nutrient deficiency or soil fertility. And biological test, using a microorganisms, we can find out the indirectly find out the soil fertility level, and the direct indication through soil test and a field experiment. So among all, the important tools are soil testing and plant analyzing. Most of our soil science laboratory is concentrating on these two. But in this session, we are going to see only about visual method of diagnosis and analysis of plant. The first evaluation method is nutrient deficiency symptoms. So all kind of plants which will indicate the deficiency symptoms, specific deficiency symptoms. So by visually we can identify the plant nutrient deficiency symptoms, because the plant is will indicate us. There are certain indicator plants which will indicates very clearly about the nutrient deficiency. So we have to take some precaution in interpreting deficiency symptoms. Symptoms may be caused by more than one nutrient. So sometimes you cannot differentiate the nutrient deficiency symptoms exactly, because some multiple nutrients might have produced the deficiency symptoms. You should know how to differentiate this. The deficiency of one nutrient may be related to excessive quantity of another, means antagonistic effect of nutrient. For example, phosphorus, zinc, and both are antagonistic in nature. So when the phosphorus content is increasing, zinc content will decrease. So both vice versa will happen. So you have to identify this. And difficult to distinguish between deficiency symptoms and the damage caused by insect and disease. Sometimes the insect affected area and diseased area in the crop plants sometimes may be shown like deficiency symptoms. So we have to carefully identify or differentiate the deficiency symptom due to nutrient deficiency, and symptom may be caused by more than one factor. Okay, not only through nutrient deficiency, some other factor also involved here, and the symptom may appear. Abbreviate outline of visual symptom of nutrient deficiency. See here, they have indicated the type of nutrient, and in this side they have shown how the deficiency symptoms will appear. First one is nitrogen, chlorosis of tips and the whole leaf stunted. So if this is the leaf, the deficiency symptoms will start from the tip of the leaf. So it will comes like this, and finally it will show like a V shape symptom, V shape chlorosis. and it starts from plant leaf tip and coming towards the petiole in this area. So it will be observed like a inverted V shape. Second, phosphorus. Phosphorus deficiency symptoms is very unique. They it will produce a reddish purple color on the leaves in all the crops, reddish purple colors, and also stunted growth because it is very important for the root development. And the next one is potassium. Potassium, always the leaf margin will get scorched. The margin, the dried leaf margin is the typical deficiency symptoms of potassium. uh, and the next one is magnesium. Spotty and slender streaks of intervenal chlorosis. Intervenal chlorosis is the deficiency symptoms, and also the leaf tips will be cupped, means curled upward curling is the deficiency symptoms of magnesium. And especially in cotton, it will produce the reddening. The leaf part will show the leaf area completely turn into reddish color. It is not a phosphorus deficiency. And zinc mainly the affect the internode development. The shortened internode is the deficiency symptom of this zinc. And also the leaves are clubbed together, that is called rosettes. Rosettes of leaf is the deficiency symptoms of zinc. The next is calcium and boron. They are immobile nutrient. Always they are showing a deficiency symptoms on the growing tip of the plant. Terminal bud dies or deformed. Terminal bud, the grown bud or growing tip of the leaf will get dried. Then sulfur. Overall chlorosis of leaves, especially the tea is an indicator plant where you can see a clear deficiency symptoms of sulfur. Complete leaf will turn into yellow color. And manganese, dead spot scattered in leaves. The spots will be appear. Dead spots will be present in the entire leaves. Veins alone green color. And it is also producing a deficiency symptoms in variety of the plant, manganese. And iron is very important element which will often produce the deficiency symptom of intervenal chlorosis. Intervenous chlorosis, sometimes the complete loss of chlorophyll will happen and the leaf will turn to white in color. So sometimes the lime calcium carbonate present in the soil will induce the deficiency symptom of iron. That deficiency symptom is called iron induced chlorosis. And copper, leaves permanently united, maybe some chlorosis. A wart like a wart like production will be there in the fruits, especially in the citrus fruit. If you see, there are some what are spots, brown color spots will be appear. That is due to copper deficiency. Leaves permanently united. So this is the another symptom of copper deficiency. So you can see here hidden hunger. It refers to the situation in which crop needs more of a given nutrient without showing deficiency symptoms. The plant will not show any deficiency symptoms, but the crop needs more amount of nutrient. More amount of nutrient. This is called hidden. Hidden, nothing but hiding. The hunger is hiding. Okay, the plant will not show the deficiency symptoms. Deficiency symptom is hiding, but the plant needs more amount of nutrient. You can see this graph showing the yield. The yield is starting from lowest and it is slowly increasing, and this part is called physiologically optimum yield. The plant should attain this yield, but the symptoms will show when the nutrient supply is reduced. Okay, at the same time, the nutrient is needed for the plant, but it is not showing any deficiency symptoms. So it will not attain the maximum limit. It will limits to in this range, that is called hidden hunger. Again, the plant shows no obvious symptoms, yet the nutrient content is not sufficient to give the profitable yield. Okay, this is what the hidden hunger. So it is a more valuable to identify hidden hunger. Hidden hunger will not show other deficiency symptoms. You can find out nitrogen, phosphorus, potassium, but the hidden hunger will not show any deficiency symptoms. For that, the plant analysis is required. Analysis of tissue from the plant. So tissue analysis, instead of collecting the whole plant and doing the analysis for different parts, certain plant parts can be obtained from the field and as a fresh tissue, the fresh tissue uh, nutrient range can be analyzed. Okay, so two general types of analysis. One is test on fresh tissue in the field and tissue analysis performed in the laboratory. So in the growing plant we can collect the samples like cell sap, the liquid portion of the plant you can collect, and the nutrient can be analyzed. Okay, instantly, immediately it will give the result. The second one is analysis performed in the laboratory. So after bringing into the lab, there will be some processing steps. After that, you will make some analysis on the plant samples. So first one is tissue test. We will see. So this tissue test, otherwise called green tissue test, we because we are not going to bring into laboratory and not going to make any drying process. So this green tissue test determine the nutrient shortages before they appear as symptoms. Anytime you can make this kind of analysis. No need to be wait until the deficiency symptom is shown. So you can collect even after 15 days of sowing or 20 days, 25 days, 30 days of sowing. You can collect the plant samples and fresh tissue analysis you can make. So this aid in determine the nutrient supplying capacity of the soil. So it is the indirect indicator of soil nutrient, the soil is supplying enough nutrient or not. This kind of conclusion you can make here. And the effect of fertility treatment on nutrient supply. So if you have done any fertility management, what is the effect? Okay, previously you would have got some deficiency symptoms, lost some earnings. So according to that, you might have added some fertilizer or any manure. So that treatment, how it is works. Okay, how the nutrient supplying power is improved in the soil, and to study the relationship between the nutrient status of the plant and crop performance. So nutrient status of the plant as well as the crop performance you can monitor using this tissue test, green tissue test analysis. Here this is the procedure. You can see rapid test for determination of nutrient. So instead of waiting a couple of days or 1 week, or couple of days to 1 week for the whole plant test, immediate result you will get through the rapid test analysis. Concentration of nutrients in the cell sap you will identify. The plant parts may be chopped up and extracted with the reagents. So the plant sample is collected, and these plant samples can be cutted into small parts, then extracted with reagent. The plant sap or cell sap can be extracted using a suitable solution. And in rapid test, the plant tissue is squeezed with plier. This is called plier. Using this plier, when you place the sample here and pressing the handle, it will give some liquid substance. The liquid substance can be placed in the port showing here. Okay, so after that it will show the reading here, or else where you can compare the color change when you are adding any salt or any chemicals to the solution. Color change will occur. So that color change you can compare with the standard color chart. And this is the easy method to conduct and interpret. And this result always indicate the range of supply and can be read very low, medium, or high. You cannot find out the exact quantity of nutrient, but quality wise you can say whether it is low, medium, or high. Example, you can see the particular growth period you can collect the plant sample, specific plant parts, for example leaf alone, petiole along with the leaf, and stem part, or root age root part, or the growing point. Okay, any part you can collect based on the type of plant. For example, you can take the maize, corn in flowering stage, base of stock for nitrate. The stock base you have to choose for tissue testing. And mid-rib, first mature leaf for phosphate and potassium. For phosphate analysis, mid-rib of the leaf, mid-rib, this is called mid-rib, the middle vein is mid-rib. And for potassium, first mature leaf, first fully opened leaf is that you have to take for tissue test. And soybean, in mid-season you have to collect the pulvinus, first mature leaf for sulfate, and petiole, first mature leaf for potassium. Likewise, cotton, in the early bloom stage you can collect the sample, the petiole basal leaf for nitrate, phosphate, and potassium. For all the things, all the analysis, petiole basal sample you have to collect. This is the picture shows the sample is being kept inside the plier. Once you squeeze this plier, you can collect some liquid here, and that liquid can be placed here directly, it will show. Or else when you are adding any uh, suitable chemical, the color change will occur, and based on the color change you can find out the type of concentration of the new. So in general, latest mature leaf is used for testing, while immature leaves at the top of the plant are avoided. So always you have to choose a fully opened latest mature leaf. So that is called most recently matured leaf, MRMIL. This leaf you have to choose always instead of collecting a recently opened leaf. Another important thing is plant pots. Plant pots and the time of testing are important. At what time you have to collect the plant sample? Like the sample must be collected from 10 to 15 places, and the average value should be considered. So 15 places, 10 to 15 places you are taking in 1 acre of land. In another acre, 10 to 15. Likewise, you have to collect. Okay, in a zigzag pattern, you have to find out you have to choose the plant and find out collect the samples. It is also advisable that test should neither be made so early nor so late in the evening. So it will change the nitrate content usually in both the times. Okay, so early in the morning and so late in the evening you should not do the plant sample analysis. That will affect the nutrient concentration. The analytical method for green tissues. Here you can see there are two main methods mainly employed for analysis of green plant tissue. One is paper test, another one is glass vial test. The material and equipment required for making tissue test for NPK are test papers, for potassium test paper, nitrate powder, sharp knife, needle nose pliers. Pliers, here also you have to take. The potassium test paper contains 3 spots of different amount of dipicrylamine. 3 spots, orange color, 3 spots will be there. When you are placing the sample, the color change will occur depending on the nutrient content. The nitrate nitrogen test is made by placing the cut portion of green plant tissue on a clear portion of the folded tissue paper, adding NO3 powder to the tissue and squeezing the paper and tissue together with the plier. So what you have to do, you have to cut the uh, portion of plant. That green tissue will appear here on the cutted area. Then the tissue paper has to be placed on here and folded. When you are squeezing out, the solution will comes out, means the tissue uh, juice will comes out. Then the color of the nitrate containing powder will be there along with the tissue paper. So when you are squeezing this plant sample with the tissue paper, the color change will appear. The shades of red will show nitrate nitrogen is present. If the color change to red color, nitrate nitrogen. Faint pink color, which indicates low or deficient amount of nitrate. A complete red color of the powder indicates high amount of nitrogen. So when you are placing the tissue paper with the nitrate powder along with the plant sample, so the paper should convert to complete red color. If it is showing very lowest shade of red color, nitrate nitrogen just is present, but complete red color will indicate a sufficient amount of nitrogen is present. But the pink color should not appear because it will indicate the lower amount of nitrogen. Then for phosphate, the phosphorus uh, 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 available phosphorus available to low, medium, or high. You can see again squeezing the cells up from the plant, and on the paper strips you have to keep an adequate supply of phosphorus indicated by medium blue to dark blue color. So this portion is adequate supply of nutrient, and these shades showing very light blue shades, so which indicate phosphorus deficiency, and you have to add some phosphorus input immediately. For potassium test, the plant sap is squeezed on 3 test spots containing dipycrylamine and reagent number 1. Okay, the when you added the reagent number 1, given orange color present. Okay, the intensity in the brightness of color determine the amount of potassium. So once the disappearance of orange color from all 3 spots, the test paper indicates very low. So 3 spots are there. If the orange color will appear after adding dipycrylamine and reagent number 1. When you are squeezing the plant sample, if the orange color is disappeared, very low amount of potassium. So orange in the middle and bottom spots alone, middle and bottom spots alone indicate medium potassium. When the orange color persists in the all 3 spots, first one, middle one, and bottom one, potassium level is high and sufficient. So when you are squeezing your plant sample, the orange color spots should not disappear. The next analysis is total analysis, performed on the whole plant on a specific plant parts. Whole plant you have to collect from the field. So this plant bring into the laboratory, and it will be washed for any sand material, on any polluted dust material, anything. If it is there, it has to be washed in the running tap water. Then it will be allowed for shade drying, then converting into a finest particle by grinding, then digested with sulfuric acid, sulfuric acid or triacid. Triacid is a triple acid extract in the ratio of 9:2:1, or else diacid. The diacid is employed for all the nutrient except nitrogen. But the triacid is only all kind of nutrient you can analyze using triacid. Then plant sampling guidelines also you have to follow. In what time, what part of the plant, and which stage of the plant, and uh, which time you have to collect the plant samples, everything you have to understand. So for that, separate guidelines are available. And the critical nutrient concentration. So this is the approach which is commonly used in interpreting plant analysis result and diagnostic nutrient problem. So this is the critical nutrient limit, which is the optimum level of nutrient. When the nutrient fall below this optimum level, definitely shows the deficiency symptoms. When it is increasing, it will attain the economical yield. So this CNC, critical nutrient concentration will determine whether the plant will shortfall or increase the nutrient concentration. See, you can see here the relationship between nutrient concentration and the plant growth. You can see the different stages of plant growth will be there. The plant starts from the low and slowly increasing and attaining a sufficient range. Okay, so this point is called critical range. It is best between the lowest part and the upper sufficient level part. Once the critical nutrient is crossed, the plant will show the desired yield or required yield. When the plant is struggling below the critical nutrient concentration, it will definitely show the deficiency symptoms. At the same time, when the nutrient is uptaking by the crops in a higher concentration, definitely it will also cause the deficiency, sorry, reduction in yield due to toxicity of the element. And luxury consumption which is associated with the potassium element, it is nothing but the potassium required for the optimum yield. So the plant will consume excess amount of potassium rather than required. Okay. So this consumption, this is the potassium, optimum potassium is required, but the plant absorb excess amount of potassium. The excess amount difference is called luxurious consumption, mainly associated with the potassium deficiency, potassium element. So this is the different element and general range and critical level. General range of nitrogen should be 2 to 4. The critical range or critical nutrient concentration is less than 2. When the plants fall less than 2 percentage of nitrogen, definitely it will show the deficiency symptoms. The range should be 2 to 4. So similarly, for phosphorus 0.2 to 0.5, potassium 1.5 to 3, and for other nutrient also like micronutrient given in ppm and critical level given in percentage. So definitely the plant should have this much percentage, like 5 ppm, 4 ppm, 15 ppm of nutrient. With respect to the element, it will differ. So it is very important to maintain the nutrient concentration in the plant. So these all about the soil fertility evaluation through tissue test or the whole plant analysis. Thank you.